viewers, today we learn blood group typing. Discovery: the ABO blood group system is the most important blood group system in human blood transfusion. The associated anti-A and anti-B antibodies are usually IgM antibodies, which are produced in the first year of life by sensitization to environmental substances such as food, bacteria and viruses. The ABO blood types are also present in some other animals, for example, rodents and apes such as chimpanzees, baboons and gorillas. It was in 1901 that Australian American immunologist and pathologist Karl Landsteiner discovered human blood groups. His work helps to determine blood groups and thus opened a way for blood transfusions which can be carried out safely. In 1902, the fourth main type, AB, was found by De Castro and Sterling. It was the observation of Levine and Stinson in 1939 and Landsteiner and Wiener in 1940 that laid the foundation of our knowledge about the remaining major blood group, that is the rhesus, that is Rh system. For this discovery, Landsteiner was awarded the Nobel Prize in Physiology or Medicine in 1930. Principle: the ABO antigens are primarily glycolipids which are found on the surface of human red blood cells as well as in association with other body tissues. These are two distinct parts to ABO grouping. The direct or forward grouping requires known anti-A and anti-B typing antiserums for testing unknown cells. The indirect, that is reverse or back grouping, requires a pool of known A and known group B cells. Both forward and reverse grouping is routinely carried out. In forward grouping, the monoclonal antibodies are available for A, B and Rh antigen. Monoclonal antibody against antigen A, also called anti-A, the monoclonal suspension being blue in color. Anti-B comes in yellow color. Anti-D, that is monoclonal antibody against Rh, is colorless. All the color codes are universal standards. When the monoclonal antibodies are added one by one to wells that contain the test sample, that is blood from patient, if the RBCs in that particular sample carry the corresponding antigen, clumps can be observed. The forward typing antigen-antibody reaction results in visible agglutination of the red blood cells, determining the blood groups A and B and AB. No agglutination with anti-A, anti-B determines the amorphous group O. The approximate frequency of each antigen on the erythrocytes of a Caucasian population is for A 41%, for B 9%, for AB 4% and for O 46%. The blood types. The ABO blood typing system groups the individual blood into one of the four categories. Blood group AB individuals have both A and B antigens on the surface of their RBCs and their blood plasma does not contain any antibodies against either A or B antigen. Therefore, an individual with type AB blood can receive blood from any group, that is with AB being preferable, but cannot donate blood to any group other than AB. They are known as universal recipient. Blood group A individuals have the antigen on the surface of their RBCs and blood serum containing IgM antibodies against the B antigen. Therefore, a group A individual can receive blood only from individuals of group A or O, with A being preferable, and can donate blood to individuals with type A or AB. Blood group B individuals have the B antigen on the surface of their RBCs and blood serum containing IgM antibodies against the A antigen. Therefore, a group B individual can receive blood only from individuals of groups B or O, with B being preferable, and can donate blood to individuals with type B or AB. Blood group O individuals do not have either A or B antigens on the surface of their RBCs, and their blood serum contains IgM anti-A and anti-B antibodies against the A and B blood group antigens. Therefore, a group O individual can receive blood only from a group O individual, but can donate blood to individuals of any ABO blood group, that is A, B, O and AB. Then there is the Rh factor blood grouping system. Rh positive individuals with Rh positive blood have Rh antigens on the surface of their red blood cells. Individual with Rh positive blood can receive Rh positive or Rh negative blood. Rh negative individual with Rh negative blood do not have Rh antigens. Individual with Rh negative blood can receive only blood that is also Rh negative. Together, the ABO and Rh grouping systems yield the complete blood type. There are 8 possible types: O positive, O negative, A positive, A negative, B positive, B negative, AB positive and AB negative. With this information, we will go for practical demonstration. Materials required: sterilized lancet or needle, microscope slides, ethanol, cotton, anti-A, anti-B and anti-D sera. Procedure: obtain a clean microscope slide. Using a marker, mark as A, B and D on a slide. Lance the finger to obtain blood. Clean the palmar surface of the third or fourth finger with a sterile lancet or needle. Make a puncture on a fingertip. Wipe off the first drop. Massage the finger from the bottom to the top to encourage blood flow. Press the blood towards fingertip. Place 1 drop of blood on each of the marked slide. Add 1 drop of anti-A serum to the A side. Add 1 drop of anti-B serum to the B side and 1 drop of anti-D to D side. Spread each mixture over an area of about 0.75 in diameter. Observe the slide for any agglutination of red cells. If agglutination occurs on side A only, individual have the blood type A. If it is occurs on side B only, individual have blood type B. If a reaction occurs on both sides, individual have type AB. If no reaction occurs on either side, individual have type O. If agglutination occurs on side D, then individual have blood type positive for respective blood group, and if no agglutination occurs on side D, then individual have blood type negative for respective blood groups. Observation: agglutination occurs on side B and D. Results: blood group is determined to be B positive by the direct or forward blood group typing method. Significance of blood group typing: ABO grouping is required for all of the following individuals: blood donors and transfusion recipients. Blood typing is done prior to a blood transfusion or when classifying an individual's blood for donation. If individuals are given incompatible blood, it can lead to blood clumping or agglutination, which can be fatal. Transplant candidates and donors: ABO antigens are found in other tissues as well. Therefore, the transplant candidates and donors must be compatible. Paternity testing: since the inheritance of the ABO blood group system is very specific, this serves as one of the first methods to determine the likelihood that the accused father is the father or not. The major importance of the RH system for human health is to avoid the danger of RH incompatibility between mother and fetus. Blood typing is especially important for pregnant women. If the mother is RH negative and the father is RH positive, when first time an RH negative woman becomes pregnant, there usually are not incompatible difficulties for her RH positive fetus. However, the second and the subsequent births are likely to have life-threatening problems for RH positive fetuses. The risk increases with each birth. Normally, anti-RH positive antibodies do not exist in the first time mother unless she has previously come in contact with RH positive blood. Therefore, her antibodies are not likely to agglutinate the red blood cells of her RH positive fetus. Placental ruptures do occur normally at birth so that some fetal blood gets into the mother's system, stimulating the development of antibodies to RH positive blood antigens. As little as 1 drop of fetal blood stimulates the production of large amounts of antibodies. When the next pregnancy occurs, a transfer of antibodies from the mother's system takes place across the placental boundary into the fetus. The anti-RH positive antibodies that she now produces react with the fetal blood, causing many of its red cells to burst or agglutinate. As a result, the newborn baby may have a life-threatening anemia because of a lack of oxygen in the blood. The baby also usually is jaundiced, fevered, quite swollen and has an enlarged liver and spleen. This condition is called erythroblastosis fetalis. It can be prevented for women at high risk, that is, Rh negative women with Rh positive mates, by administering a serum RhoGAM containing anti-Rh positive antibodies into the mother around the 28th week of pregnancy and again within 72 hours after the delivery of an Rh positive baby. This must be done for the first and all subsequent pregnancies. This treatment can be 99% effective in preventing erythroblastosis fetalis. RhoGAM is also routinely given to Rh negative women after a miscarriage, an ectopic pregnancy or an induced abortion. To summarize, the ABO system is the most important blood group antigen system. Individuals who lack the A and/or B antigens on their red blood cells generally have naturally occurring antibodies in their plasma that are directed against the missing antigens. An individual's ABO group is determined by testing the red blood cells with reagent anti-A and anti-B sera. Agglutination of the individual's red cells by the appropriate anti-sera signifies the presence of the antigen on the red cell, while no agglutination with the anti-sera signifies its absence. Proper determination of the ABO group is necessary when selecting red blood cells containing products for transfusion in order to prevent immediate hemolytic transfusion reactions. The importance of Rh system in case of pregnancy is also mentioned. Thank you.